Welcome to Word of Hope, Ovarian Cancer Podcast, brought to you in partnership with Word and Libby's Hope. For more links and information on this episode, visit wordofhopepodcast.com.
Welcome to Word of Hope, Ovarian Cancer Podcast, Episode 4. Our hosts are Paul Cacciatore from Libby's Hope, and I'm Nate Manahan from Women's Oncology Research and Dialogue, and we are continuing to talk about 10 exciting ovarian cancer research topics that most were talked about in 2010. And so as we walk through these, we've gone through 5 topics. Now we're going to talk about the next 5, which are all medical discoveries, and Paul has chosen one today, the ARID1A gene mutation. Paul, why don't you start us out by talking about this first medical discovery?
Okay, and the first one is a finding of a frequent mutation in a particular gene in 2 types of endometriosis-associated ovarian cancers. The first being clear cell, the second being endometrioid ovarian cancer. Both of those are 2 subtypes of epithelial ovarian cancer. And just to kind of give you an idea, the main subtype is serous ovarian cancer. And then the next type would probably be clear cell after that, I think, running at around 12% of that remainder. This is a really interesting case, and I think maybe the best way to do this, since we'll be talking about genes, is to kind of take a step back and do a little bit of genetics 101 on this to help people understand how this works.
I think that's a great idea. For most of us, it's been a long time since high school biology. So why don't you give us a quick overview of genetics and DNA?
DNA is the recipe or the code that's contained in every human cell. And DNA contains that recipe or that formula or the blueprint, if you will, of what's needed for the cell to make a protein. And proteins, as we know, are the kind of the workhorse of the human cell. It allows the cell to carry out a whole series of functions in its everyday life. So needless to say, to have that recipe be correct is very, very important. And what happens then is the DNA contains that code. RNA, which is another term we hear, ribonucleic acid, has a messenger element that comes in to copy the recipe in the nucleus of the cell and carries it outside the nucleus of the cell, and then uses that recipe to go ahead in what's called the cytoplasm of the cell, outside the nucleus but still in the cell, to go ahead and manufacture that protein.
Now, this is reminding me of Mr. Brenneman's biology class, and my memory serves me right. There are some letters that represent that recipe. Could you tell us a little more about that recipe?
So with that recipe, it's basically, to make things simple, it's a string of letters, for kind of a lack of a more detailed description. It's biologically, it's a series of different bases, 4 in particular, A, C, G, and T. And if you can kind of picture those letters repeated over and over and over again in a string, doesn't mean a whole lot to the average person, but it means a lot in terms of how these proteins are manufactured. And snippets of that code, a portion of it is what we refer to as genes. And so if a gene is mutated or a portion of that code is in any way not correct, to give you an example, the best way I think to think of this, the way I visualize it, is sentences in English. If you leave out a word in a sentence, it doesn't seem to make much sense. If you transpose words in a sentence, it doesn't seem to make much sense. If you misspell words within the sentence, it doesn't seem to make much sense. And really, in kind of an overarching way, that's the way mutations occur. There's a whole series of different kinds of mutations, but the bottom line is the recipe as intended isn't quite right. Maybe too much sugar, too much salt, or too much pepper, to use a cooking analogy. And so that's where mutations in the genetic area become very important. The group of researchers up in British Columbia, up in Canada, and their name there, it's a large group of researchers with the Ovarian Cancer Research Program of British Columbia. And they have doctors from the BC Cancer Agency, Vancouver General Hospital, a number of different organizations up there that work cross-discipline backgrounds, all working together as one team. So you have gynecologic oncologists, pathologists, There are people there with drug development expertise, things like that.
Paul, let me pause you just for a second, because I think that's a really important point, that this research project and most of the research projects we look at are cross-discipline research. And that means there's multiple specialties, like you said, working together to get to the point. And I think that's really important for people to look at when they're looking at and analyzing research projects, is that's not just one viewpoint coming, it's multiple viewpoints all working together to make sure they're coming to good conclusions. Now let's look right back at this research project. This is really interesting, Paul.
So what they did is they looked at these clear cell and endometrioid tumors. And what they found out was that about 1/2 of the clear cell tumors and about 1/3 of the endometrioid tumors possessed a mutation in a gene that's called ARID1A. It's ARID1A. And the reason that's important is that this gene, and remember, genes encode proteins, or they have the recipe for the protein. The protein that's associated with this gene is called BAF250A. The name of it isn't as important as is its function, and it's a key component of a cellular structure that has actually a lot of functions that it does. It's involved with development, differentiation, proliferation of the cell. It helps with DNA repair if the cell gets damaged. It helps with tumor suppression if a cell gets out of control. And so there's a lot of important functions associated with this. So if you can kind of understand this lineage, you have the gene that's mutated. The gene is responsible for coding the protein. Well, a protein's only as good as what it's made from, or the recipe it's made from. And if the recipe is damaged in any way or incorrect, you're going to have basically a bad protein that may cause things that we don't expect within the cell.
Wow, Paul, that was like a whole semester of genetics in just a couple of minutes. That's a great description. Now let's talk more specifically about this ARID1A mutation.
So this ARID1A gene was found to be mutated, like I said, in about 50% of clear cell cases and about 1/3 of ovarian cancer cases. What they did is in their study, they went through a number of, and I won't get into all this detail because it's fairly complicated, but what they did is they checked it first at the gene level. They found the mutation. What they then wanted to know was, okay, this associated protein, do these things work in tandem? Whenever you see a gene mutation in ARID1A, do we also see problems with the protein? And in fact, they did. And actually in the serous ovarian cancer tumors that were part of this, the research that they did not find this type of mutation in serous ovarian cancer, which is the most common kind of epithelial ovarian cancer. And so, it's interesting from that standpoint. What they concluded is they do believe that ARID1A is a tumor suppressor gene. One of its primary functions is to kind of keep the cell in check and to prevent the cell from, multiplying rapidly in an uncontrolled manner. And they think that it's a tumor suppressor gene and that this mutation is a defect in a very important gene that would help, to stop cancer, in the beginning. And so that was a very important finding. They also found mutations in several other genes, KRAS, PIK3CA, CTNNB1, again, all names that may not be familiar to the layperson. But the interesting thing about those mutations is they had been found by other researchers before this. So ARID1A it's a novel finding. It was reported in the New England Journal of Medicine last year. And I should also note that Johns Hopkins, on the same day this study was published in the New England Journal of Medicine, Johns Hopkins also published I don't want to say a similar study. It's on the same subject matter of ARID1A in Science magazine.
That's great to have two studies on the same genes. What were some of the findings from Johns Hopkins?
What they found out, too, was that much like the researchers in British Columbia, they found out that the ARID1A mutation, they identified it in 50% of the tumors that they checked, the clear cell tumors. So that's good because you had two separate groups working on a similar issue. Both came to the same conclusion and to a similar finding.
Paul, what is the takeaway message of this findings, and why is it really important for ovarian cancer research?
I think the takeaway from this is why this is significant is much of the research, the drug development that's gone on, looks for targets. It looks for therapeutic targets that they can use to kind of to keep the cancer in check or to destroy it. And this is a good isolated gene mutation that may be susceptible in some way to a therapy down the road. And in fact, the researchers at British Columbia have a multi-year project already going on where that is exactly what they're going to do with this ARID1A gene. They're going to look at it in more detail. They're going to check for other genes that may follow suit. And with the hope that they can develop from that information a specifically tailored drug that can be used to fight this specific form of ovarian cancer. The second thing that it also does is that the science over time, and this British Columbia group was instrumental in demonstrating this point back in 2008, that ovarian cancer isn't one disease. It's a grouping of distinct diseases. So under, for instance, epithelial ovarian cancer, there's 4 subtypes. You have serous, clear cell, mucinous, and endometrioid. What this suggests is that each one needs to be looked at on its own, and that there may very well be therapeutic approaches down the road here unique to that particular form of ovarian cancer. And so I think that's the takeaway point, is that the science is moving in that direction. It's not going to be a one-size-fits-all type treatment. Rather, they're going to look for specific flaws or defects in the ovarian cancer cell, sometimes unique to a subtype of ovarian cancer, that can be exploited through drug development, and so that it will help stop the cancer down the road. So I think that where the science is going is they're trying to identify defects in particular subtypes of ovarian cancer, rather than having all these women treated with, for instance, one standard of care down the road. Because when you think about it, it makes a lot of sense, because there's a lot of women that will go on a particular drug and have a great effect, great benefit, and there'll be some women that will go on the drug and have no benefit, which strongly suggests that even from person to person, regardless of subtype, the cancer could be that unique, from person to person. So,
Paul, I think this is great, and I think this really emphasizes one of our key messages for ovarian cancer patients. Is make sure you know what type and subtype of ovarian cancer you have. And specifically, this will influence what kind of treatments you may be able to have in the future. And it may not have an impact today, but it may have an impact in the near future as they develop new treatments for your subtype of cancer.
That's right. No, that you raise a really important point. And I think one of the much overlooked documents for a patient is, in fact, the pathology report. Yeah. We've always known that pathology is important. And I can tell you, Nate, as time goes forward and the science becomes more developed, knowledge of your pathology report becomes critical, I think. Yeah. Because what it's doing is it's telling you what your particular form of cancer looks like. What are the characteristics of your particular form of cancer? And just give you an example within clear cell ovarian cancer. They know, for instance, that clear cell tends to have a mutation in the PIK3CA gene. And right now in clinical trials, there are PI3K inhibitors. So see, there's an example where it's not a perfect fit and it's not, perfectly reliable, but we are starting to get to the point where we have some ability within clinical trials to try to match a drug to a particular characteristic of a subtype of ovarian cancer. Yeah, so I think that's again where the significance comes from. I get a lot of inquiries from women, and the first thing I usually ask them is if they did get a copy of their pathology report. And more times, more often than not, they have not obtained a copy of it. And so this is just a great example of once you're diagnosed, having the ability to stay organized and to make sure you get copies, and not just of your pathology report, as you know, but your surgical report, testing, blood testing results, things like that. I think it's always a good hand to kind of keep your own book, your own file folder with those tests in it. Because it helps as the science develops, you're able to go back to your own characteristics, compare it to what you're seeing reported publicly, and then you can ask your doctor intelligent questions from that.
Absolutely. Paul, this is a great discussion to have about ARID1A gene mutation and even individualized treatment and the importance of pathology and type and subtype of ovarian cancer. I hope that the viewers and listeners of this podcast will join us over at wordofhopepodcast.com. There's all kinds of links and references there, some great articles from Paul. And we hope that you'll leave some comments, questions. And if you want to learn something more or have a specific question, just hit contact us and we will be glad to answer any of those questions for you. Please join us next time for episode 5 as we talk about the reliability of CA-125 in recurrent ovarian cancer and in treatment. This will be a great discussion of its importance, and I hope that you'll join us. See you next time at the Word of Hope Ovarian Cancer Podcast.
You've been listening to Word of Hope Ovarian Cancer Podcast, brought to you in partnership with Word and Libby's Hope. For more information on this episode and how to subscribe, visit wordofhopepodcast.com.